And he'll be presenting research on optochemical continuous monitoring of glucose and whole blood for ICU and CCU setup. And your research collaborator is?
So one of them is a PhD student, and he may have graduated, but his name is Stovik.
Yeah.
First name Stovik.
All right.
Okay. So for to keep the timing so that it's like under 10 minutes, I'm just gonna go through most of these slides very quickly. Some of them I'll skip, and if you have questions, I can go back to them. This is just an overview of what I'll be discussing. So this sensor that we developed through our research is something that measures the concentration of glucose in blood, and we are specifically looking for the glycemic variability, GB, which is essentially how the concentration changes over time. And we're doing that specifically in blood, and it's used in ICU or CCU setup specifically. I got you. So I'll be skipping this slide. I may come back to it at that time. All right, so these are the problems that our group identified for our glucose sensor in blood. So specifically, we wanted to create a sensor that does not have blood fouling. So if blood cells, if they go onto the surface of the sensor, they deposit like red blood cells, white blood cells, larger proteins. That can reduce the sensitivity in the long run. That can make it less reusable, it can be less stable. And the last challenge that we tried to overcome is we wanted to make our sensor that it can monitor glucose level continuously in real time and have access to that information without losing that. And then we identified certain solutions that we did and were successful when we developed our sensor. And so one of them is that it's portable. Another is that it is minimally invasive to the patient. We're using a catheter, which is something that's existing technology. So simply we just insert our sensor to the catheter that draws the blood, that gets the information. We do signal processing, and that's what we use to determine the glucose concentration. And then another part is, of course, we need to protect the sensor from blood fouling, as I mentioned earlier. So we need to have some kind of mechanism for that, which I will discuss later. And then also, it obviously has to be continuously monitoring blood continuously, which is the most important part of our sensor. All right, so this is the diagram showing our sensor. So there's the body, and we use a catheter. Inserted into the catheter is our microdialysis probe, and then connected to the probe is our sensor, optical fiber sensors, connected to a detector that detects the signals, and then that's connected to a computer that can do the processing, signal processing, and analysis. And then, so this is our objective, I already explained that earlier. Then our sensor has 3 layers. So the first is the core, which is silica-based. Then on top of that is the gold nanoparticles. And the purpose of that is to actually get the signal, the LSPR signals. If I have time, I can explain that more in detail. And then on top of that is the final layer, is the glucose oxidase layer. And that is actually responsible for detecting all the glucose molecules. So as that concentration changes, glucose oxidase can detect the change. And glucose and the combination with glucose oxidase, which is the enzyme, which acts as a catalyst for the reaction, that produces certain byproducts. And when those byproducts, that changes over time as the glucose concentration changes, you will see a change in the local refractive index at the sensor surface, and that causes a change in the intensity when light is sent to the sensor surface. So as the concentration of glucose changes, that changes the local refractive index, so it changes the intensity at the LSPR wavelength. And by checking both the true concentration of glucose in blood over time at different intervals, and also looking at the sensor response, we can see the relationship and use that to determine the concentration of glucose. And I will skip this. This is just how the sensor is fabricated for purpose of saving time. And then this is a setup for in vitro, which we did in a phosphate buffer solution. So in comes the glucose through the flow cell out to the outlet. Then we have the sensors where all the readings are there. And then we have a light source where light travels through the wire, gets to the sensor at the distal end where the optical fibers are located. And then that is reflected to the detector where the signals can be detected. All right, this is the setup in vivo for this. This is in a live rat experiment which we did at the University of Iowa. So this is a circuit showing how the blood flows during a surgery and then comes back to the rat. So it goes through this probe, which has a 20 kDa cutoff. So anything larger than that, then it would just simply go back to the rat. Anything smaller than that, including glucose, goes to the sensors at the outlet, and that's where all the glucose oxidase is, where the reaction undergoes, and we see the change that we can detect. There's also this inlet where perfusion fluid is inserted that contains an anticoagulant called heparin that is used as to prevent blood clotting on the surface of the microcystic dialysis probe, so that helps with the fluidity and the diffusion rate, so it's much easier. So there is a concentration gradient as higher concentration of glucose flows from the rat side to the sensor side, and we need to maintain that, and that's why we're using an anticoagulant here in our design. And at the same time, we are tracking the true concentration of glucose throughout the whole time. So every so often, we would measure the true glucose concentration, and the whole time we are measuring the sensor response, and we're just monitoring the two variables to see what the correlation is. And I can show that in the next slide. So here's the correlation. So over 2 hours, our sensor works in single probe. Concentration glucose varied. We're measuring every so often. And the sensor response, the reflection percentage varies. So you can see the correlation is a pretty good correlation. Then again, we have a calibration curve showing the change in glucose concentration, relating that to the mean intensity, which is something that we could calculate based on the reflection percentage data. So you can see it's a very good correlation, and it's linear plots. And so the slope of this line, that's the sensitivity, and based on the standard deviation, we can also determine the limit of detection, LOD, and that is around 50-something milligrams per DL over here. And this is our performance comparison to other works similar to our work. So as I said, our work is both in vitro and in vivo continuous monitoring of glucose in blood. It's integratable with current technology using a catheter. It can work for 2 hours continuously using only one probe. It's much more simple to make and produce, fabricate. If I have more time, I can explain how that is done. And it's much more, less expensive, which is always good. And this is, if you want to see the paper, this is the paper we published. It's a link to that paper. All right, that's